Hi, my name is Randall Ray Patterson, and this is my presentation on signal transduction through Grand Canyon University, Biochemistry 530, August 2nd, 2020, Dr. Barrera. I did my project on receptor tyrosine kinases. And receptor tyrosine kinases are a large class of receptors with cytoplasmic kinase activity. RTKs, as they're called, use a pathway called mitogen activated protein and they like to activate RAS, which increases the TFs for cell division. RTKs are also involved in cancers, and this lesson that we're going to talk about today, we'll talk about how they develop and some of the ways that they can treat RTKs while in cancer. RTKs are what is needed for signal transduction pathways to conduct cell-to-cell communication. These are the types of receptors that are responsible for the binding of polypeptide ligands, and these RTKs are what is being studied for intervention to study RTK expression, ligand activation, and downstream signaling. These are a few of the things that we're also going to look at during this presentation. Receptor tyrosine kinases make up the largest class of receptors that double as enzymes and bind a signaling molecule to the membrane receptor. This allows the receptor to start an enzymatic activity for receptor activities also. The role receptor tyrosine kinases plays is that of cell growth, survival, and differentiation. When the molecules bind to a receptor, tyrosine kinase forms what is called a cross-linked dimer. This cross-linking is called cross-phosphorylation and occurs when the RTK is activated. And when the cross-phosphorylation occurs, RTK has tails on the end, and they serve as the docking platform for this protein. Once the proteins dock here, it is called signal transduction. Once signal transduction occurs, these proteins called SH2 like to bind to the phosphorylated tyrosines at these tails. And once they become activated, there can be more than one type of SH2 binding together, and this allows for more than one type of signaling pathway to occur. And once the RTK is activated, the gene transcription is changed as the signal travels from membrane to the nucleus of the cell. All right, and once the RTK is activated, it starts a signaling pathway called mitogen activated protein, or as they like to say, MAP, kinase cascade. It is called this because it involves three kinases. Once this pathway has begun, it starts by activating an RAS, which is a G protein that is found in the plasma membrane. Then the RAS likes to bind with the GTP, and it becomes an active protein. Once this occurs, the first serine threonine kinase is activated. Then after the first series kinase is activated, the other two also get activated by this phosphorylating it. They also get more amplified as they move along the kinase. So the further down they go, the more active they like to get. Then at the first step in the change of gene transcription leads to many growth factors like nerve and platelets growth factors. The nerve growth factor uses insulin like growth factor to activate enzyme phosphodieside. All right, they use this to get information to the nucleus of each of the cells. Some RTKs use transcriptional regulators or STAT proteins to get this information moved along to the nucleus. Key is getting it towards the nucleus best they can. And once this occurs, there is a change in the transcription as the information is moved into the nucleus. And for higher level functioning to occur, there must be cell-to-cell interactions. For surface proteins called ephrins, when they bind to the RTK, they like to help guide the process of developmental tissue, nerve endings, and blood vessel maturation. If the RTK does not function properly, then this can cause cell growth to go absolutely crazy. And if this happens, this is when you start to get your cancers and other various problems can occur, but mostly cancers can occur from these mutations. And we're going to talk more about the cancer formations later on in the presentation. RTKs have an extracellular domain that contains a ligand binding site, a single hydrophilic transmembrane, and a cytosolic domain. This binding of a ligand causes this dimerizing of the RTK, which leads to a process called autophosphorylation. Now, this process happens in two various stages. The first stage is where ATP is binded at the insulin receptor, and binding here can occur at the FGF receptors. Once this occurs, the receptor kinase activity occurs at other sites within the cytosine domain. For autophosphorylation to occur, there must be a binding of ligand occurring. Without this ligand binding here, then this process, it can't happen. So it's very important that this takes place. And when this takes place, it also activates the kinase at this time. And once this RTK is activated, phosphorylation residues get along with adaptor proteins. These proteins couple with the activated receptors. All right. Once this takes place, then we get into the RAS. RAS is a key signaling molecule that is triggered by the activation of the RTKs. RAS is a GTP binding switch protein that moves back and forth between a bound GTP and a bound GDP. To activate RAS, it must be triggered by a hormone that binds to a cell surface receptor. And to accelerate the RAS, a protein called guanine nucleotide likes to bind to this RAS, causing the bound GDP to break apart. After this occurs, then the GTP becomes present in higher concentrations, and they like to bind to the empty RAS molecules. All right, then these molecules become deactivated by the hydrolysis of the bound GTP. All right, this is important. The GTP only lives for about 1 minute. That's it, just 1 minute, real quick. And this is because there is the presence of a protein in it called the GTPase activating protein, or GAP. This binds to the RAS and GTP and accelerates the GTPase activity by 100 times or more. That's a lot. There is a lot. RAS is a member of the family intracellular GTP binding switch proteins called the GTPase superfamily. The RAS proteins are involved also in the are also involved, and they regulate fusion of the vesicles within the cell. There is a thought that the structure and function of RAS could have originated during evolution. I'm not sure. I'm personally not involved in, I don't believe in the evolution, but as the scientists, they like to believe in it. And reading this article, these particular scientists think that RAS could have been around since the evolution had occurred. They believe that they think that there's a single type of signal transducing in the GTPase, and so that's what they believe, and more than likely, it probably was been around since that long. The thought is here that this gene was copied during encoding and transformed into what the cells we have today. And that's probably more like it probably started out that way, and then through time it has evolved into what we've got today. And this is what allowed this superfamily to form. And so this is what the scientists have seen. The scientists have seemed to deem the most popular theory. In this superfamily, they think that they are comprised of over 100 different types of switch proteins, and that these proteins are believed to control the body's cellular growth and metabolism. So they think that as we grow, this is what controls our growth and then our metabolism. And if we were to have a mutation, and then that could slow down our growth, our metabolism, or it could cause us to have a cancer or something like that. Earlier, we talked about the RTKs being activated by dimerization. Even though this does happen, many ligands have various strategies for activating these dimeric states. Growth hormones, along with a GH receptor and a erythropoietin (EPO), in complex with an EPO receptor, these are able to prove that they are bivalent and that one ligand is able to bind to two receptor molecules to form a ligand receptor complex. Then extra receptors allow there to be further stabilization. And the simplest way that a ligand receptor can dimerize is by homodimers. An example of a homodimer is a VEGFR, and they contain 7 domains which allow them to be able to do the ligand binding. So they're able to go in and able to bind that together. The fibroblast growth factor have over 21 growth factors in them. The crystal structures in FGF, along with the ligand binding domain in FGFR, to activate this dimer. Once the dimer is activated, it stabilizes at a secondary binding site where the FGF and D2 interact. And then for this autophosphorylation and PTK stimulation to occur, then certain forms of receptor dimers must be present for them to be activated. Otherwise, then the process stops. For them to be stabilized, there must be ligand binding to the extracellular domain. There is even a thought that active dimers are present without ligand binding, since the inhibitors are still there. So that remains to be seen. To become modular in nature, signaling proteins must contain SH2 and PTB domains. The protein molecules that are involved in cellular signaling contain anywhere from 50 to 120 amino acids. The SH2 domains like to bind to distinct amino acid sequences, while the PTB domains like to bind with the PTYR. Some of the PTB domains like to bind non-phosphorylated peptide sequences. And then the SH3 domains like to bind with the rich motif PXXP sequence. And then the WW domains like to bind to the PXPX motifs. All right. The plex strand homology has over 100 domains and likes to bind to PDDLNS and to other PL3 kinases. The FYVE domain make up the PDLNS 3P protein modules, and the PDZ bind to the hydrophobic residues. Tyrosine kinases are important in signaling and pathophysiology of cancers. While in a normal cell, tyrosine kinases are regulated, but sometimes they can mutate and lead to cancers. So in this particular, now we're going to start talking about the various cancers and how they're treated. In some cancer cells, activation can be blocked by tyrosine kinases, and scientists feel that this is a good sign for the future in cancer patients, which is really awesome in my opinion. Tyrosine kinases are a family of enzymes that catalyze phosphorylation in proteins using their ATP energy, which is very important in helping these cancer patients. This is very important in the cell being able to maintain homeostasis. When this goes awry, the signaling pathways become messed up in the cancer cells, and then you get this malfunction occurring in the signaling of these proteins, and this allows for those mutations to come in and mess things up and form the cancers or tumors or whatever the patient may have. And when the Human Genome Project was decoded, scientists found over 90 tyrosine kinases there, and they played a major role in the cancer development of those patients. This is brought about by mutations within the domain that lack an amino acid 6273, and this leads to the activation of the receptor tyrosine kinases, and which can lead to cancers such as the bladder cancers, the cervical cancers. It can also lead to tumors and things like that. But very important that you realize that this is what helps to cause those mutations. Under normal conditions, the growth factor ligands like to bind to the cellular regions outside of the RTK, and these receptors are then activated by a ligand-induced receptor. RTKs enable the trans-autophosphorylation of every TKD, which releases a auto-inhibitor. This causes the TKD to become active. All right, and then once this happens, this activates the downstream signaling protein that contain the SHT we talked about earlier. Then they like to bind to the phosphotyrosine, and downstream mediates are engaged to start the cellular signaling pathway, which is very important. During their studies, scientists have found that in the genes of encoding RTKs, there have been alterations affecting them. When the RTKs are activated in cancers, there are 4 ways that this happens. First way, the gain of function of mutations, genomic amplification, chromosomal rearrangements, and autocrine activation. First, we're going to look at is the gain of function mutations. Right, these are found in the RTK, and they usually lead to downstream signaling transduction. Here you can find driver mutations that have a growth advantage in the cell. Their driver mutations can give a scientist some better understanding of how the cancer starts and how the treatments should be. Second one that we're going to look at is the genomic amplification. This leads to an increase in the amount of receptors, which in turn causes the RTK signaling to increase and overwhelms the regulatory effects. This is mostly associated with cancer patients, especially breast cancer patients. And this is to me, that's very important. I've known many people that have had problems with had breast cancer and all, and that is so serious, and it needs to be really taken. You really take it serious. When this gene amplification occurs, it causes the number of a region of the genome to increase. They occur as double minutes, or they spread out through the genome. If you get double minutes, then you're going to get more amplification, so you're going to have more problems. This can be affected by the patient having a defect in the patient's DNA replication. But if you get this amplification, you need to get it. That needs to be seen immediately, as long as once you figure it out that you got that. Third thing we'll look at is chromosomal rearrangements. When they become identified, they lead to the formation of the tyrosine kinase fusion oncoproteins. These proteins can be targeted with a small molecule inhibitor, and people with leukemia usually have the first tyrosine kinase fusion in their body. Chromosome fuses the gene encoding to form what's called a BCR-ABL fusion oncoprotein. This targets the ABL kinase and helps to treat these patients. These rearrangements of the RTKs can lead to chimer fusion proteins, and when the RTKs are inhibited, scientists have been able to prove this as an effective strategy against RTK fusion-driven cancers, and this allows those patients to be able to maybe treat those cancers better, hopefully get them put into remission, such as the leukemia here. The last one that it talks about is autocrine activation. This means that the cells are secreting themselves. This can lead to an expansion of cloning and tumor formation. Very important to remember that forming these tumors can be very dangerous. And when the RTK is activated, it can lead to helping various cancers. Using this can also lead to studying more about cancer cells. So once scientists can figure out, what's going on, they can study and try to help prevent it from happening. And thankfully, because of this, scientists have been able to make great strides in the study of RTK signaling. They have learned more ways to target the various cancer cells in the body. And with more and more study, with all these types of cancers, as we get more, our technology gets better, our scientists get smarter. We can find hopefully more and more ways to help people that have cancer. Having my father and father-in-law die of cancer, this is something that's very important to me. It's something that I always get checked myself. And in closing, RTKs are extracellular ligand body domain. They have ATP binding sites. They play a vital role in what's going on with our bodies today. When dealing with the cancers, we must treat those very, very seriously. We must continue scientists must continue to work and try to find ways to study and help with these tyrosine kinases. They can use these various strategies that we talked about in the lesson, and I think if those things are able to be done, I think that we can find a solution to our cancers, maybe find more treatments for people and help them through this study of receptor tyrosine kinases, which is very, very important in the development and treatment of our cancer patients. Thank you for your time, and I hope you all have a blessed day. And thank you very, very much. And I thank you.